So to start with, what HIV is, it stands for Human Immunodeficiency Virus. And few people realize there are actually two different types of HIV, HIV 1 and 2. And they're actually a little bit dissimilar. They actually have some different genes in them compared from one virus to the other virus. And we'll look at that in a minute. But there is a big difference between HIV and AIDS. HIV is a virus. It's what infects you. And once it's gone through an infection and it's destroyed your immune system, that's when you're said to have AIDS. You do not become infected with AIDS. You become infected with HIV. So, where did it actually come from? Well, it's been predicted that it arrived in the 1930s, somewhere in Africa, and the first case which was really truly diagnosed was in 1959, and this was, I think it was a Norwegian sailor, or a Swedish sailor, who was identified as having this. But no one really knew about it until the early 1980s. And the first place it turned up was San Francisco. Anyone remember the media hype about it? What did they say?
You mean about the homosexual population?
Yeah. So the media portrayed it as something which only affects homosexuals, and they also portrayed it as really only affecting white gay men. And that wasn't true. It did affect heterosexuals just as much as homosexuals. It affected African Americans as much as whites and any other type of race. And so the media portrayal actually led to a belief that if you weren't a white gay man, you didn't have to worry about it. And so it actually spread it around even more in the other different races and risk groups. So the media portrayal is one of the bad things which happened here with how it was portrayed. And in San Francisco was a place where it was initially identified. It was identified to start with as a group of people with immune deficiencies. And an immune deficiency means their immune system does not function correctly. And without a functional immune system, you can't fight the organisms which you encounter every single day. And an organism where your immune system would naturally defeat it and easily defeat it would be called an opportunistic pathogen. That means if an opportunity arises, it will cause a problem for you. And there are a few different types which came here. There was one called pneumocystis, which is a fungus, and that causes pneumonia. Some people, I think it's around 10 to 30% of the people, have it in your respiratory tract normally. And for you, it won't cause you any harm, because your immune system keeps it under control. You have no idea you have it. However, if you don't have the immune system, you get pneumonia. So that was one of the first instances which started to highlight the problem. These rare infections occurring for an unknown reason. Another one was a type of cancer. Your immune system actually will defeat most types of cancer for you without you knowing. Only when the cancer can overcome the immune system are you actually diagnosed as having cancer. And one of them was called Kaposi's sarcoma. It's actually a cancer of the skin and blood vessels. And normally, your immune system will defeat that easily. It's linked to a human herpes virus, number 8. And both pneumocystis pneumonia and Kaposi's sarcoma highlighted there was a problem with people's immune system, but they didn't know why. It took a couple of years, 2 or 3 years, to actually identify if it was a virus. And then there was an argument between America and France about what to name it and who actually discovered it. Apparently, the French team truly did identify it, and they sent a sample to America, and it contaminated someone else's work, and they claimed that they identified it from that. So there's a big whole long history on that. And they were also arguing about the name of it. They had AIDS-associated retrovirus or human T-cell lymphotrophic virus. After a while, the committee which names viruses says we're going to call it HIV. So neither got their way for that one. But this did affect lots of other different populations. It wasn't just the white gay men at the time. And when you come back later to think about why it affects some populations more than others, part of it is because they thought they were immune to it to start with. And that belief went on for a number of years, possibly for over a decade. And so when we look at the statistics shortly, you'll see HIV does infect different races to a different proportion. HIV-1, this is the one which is causing a pandemic. A pandemic is a worldwide epidemic. That means lots of people are being infected and have the disease. And there were a number of theories where it came from, due to the media hype. It's a plague from God against the homosexuals. That's how the church portrayed it. Some people linked it to the polio vaccine, which was used in the 1960s. That's the oral polio vaccine. It's a live attenuated virus, and it was grown in monkey cells. And then that virus from the monkey cells was put into people to vaccinate them. But that was never, there's no documented evidence that that really is where it came from. There's a lot more evidence that says it's definitely come from chimpanzees, HIV-1, and it probably came from bushmeat. So in Africa, when times get hard, people go and hunt all the local wild animals and eat them. And that's probably where it came from. Also, close association with the chimpanzees being around them all the time. And we'll look at the routes of infection shortly. We know it's from chimpanzees because there's an equivalent virus in chimpanzees. Basically the same virus. It's called SIV-CPZ for chimpanzee. And it's genetically identical to it. So we know it definitely came from a chimpanzees. HIV-2 came from a different monkey. Came from sooty mangabees, so it's SIV-SM for the sooty mangabees. And these have identical genetic sequences, and they have the same genes. Some of the genes which control the regulation of a virus are different between HIV-1 and 2. HIV-1 or HIV-2 is only still really located wherever sooty mangabees occur. So that's kind of Western Africa where it is. Both of these are considered a zoonotic transmission. Anyone know what that means?
From animals?
Comes from animals. So they're both linked to the different types of monkeys. Interestingly, the ones in the monkeys don't cause them any harm. They don't destroy their immune system. They don't even show any signs that they're infected. But in us, the virus isn't adapted, so it causes us problems. A virus's true goal is to replicate and infect you without you ever knowing, so it can be passed on to the next person. When they cross a species barrier like this one, they're going to cause us problems. Over time, it might mutate and adapt to us so it doesn't cause us as many problems. But it's not happening for HIV at the moment. So, any questions on that part? Yeah, that would be the mad scientist one.
Way possible in your mind that this is possible?
No. I would definitely not, because it is so much identical to the ones from the sooty mangabees or the chimpanzees. There's no way it could have been created like that.
So how did they get to the humans? How did they get it?
Bushmeat is the most probable cause. Bushmeat is when they hunt the wild animals and eat them, and they don't cook them properly. So that's probably the most common route of infection. We'll look at the different routes of infection, and we'll see that this virus cannot survive outside the body for a long period of time. So it really had to come from intimate contact, and it's most likely due to eating the wild animals. Because it's pretty much through the whole population of the chimpanzees or the sooty mangabees.
So pretty much every single one of those chimps?
Most of them, yes. There's such a large proportion. Some chimps which grew up in isolated areas and never mingled with the rest of the population, they might not have it. But it's on the majority of chimpanzees will have it. And that virus which comes from them will not really infect us. The one which has infected us has adapted somewhat to us to be able to replicate in our cells. So it takes time and mutations for it to be able to cross a species barrier. It can't just instantly do it. It has to accumulate so many mutations to be able to cross a species barrier. So if we took one straight out of the chimpanzee, then depending on what mutations it has in it would depend whether it could infect us or not. Things like the influenza virus also have the same problem. Influenza's a different story. It's got 8 parts of its genome and they can all be mixed up if 2 different ones infect a cell. But unless it can bind to a receptor in our body, and then it won't be able to infect us. And the receptors in the monkeys are not identical to the receptors in us. So, when I talk about the statistics of HIV, I'm really only going to be talking about HIV-1. That's the one which is all around the world. HIV-2 is mainly just in Western Africa where the sooty manga bees occur. And HIV-2 has been said to be slightly less dangerous to us. Doesn't cause quite as many problems as HIV-1, certainly not as fast. So when we're looking at this, estimated about 33 million people in the world infected with HIV. And it's stayed at that level for the last few years. So from the early 1980s onwards, it had a big incline upwards, and now it seems to have leveled off. And actually, since 2001, there's been a 17% reduction in new HIV infections. So it seems all the programs to try and prevent transmission, advertising the use of condoms or needles, drug users, all of those methods seem to be slowing down the progression of new people being infected. And when we look at the vaccines, which have been tried, I'll come back to why it's important we keep those still going. Most of it, 2/3 of it, is in Africa. And with it being in Africa, that's a country where they can't really afford the health care, they can't afford the antiviral therapies, and and they also have other problems like malaria, might distract or weaken the immune system slightly, TB as well, tuberculosis. So they have all these other problems at the same time, which could mean it's a slightly easier chance for HIV. But that's still a little bit debatable at the moment. In America, it's just over 1 million cases in the whole of the country, and we're going to take a closer look at those statistics. But it's worth noting that when we look at the HIV-1 for all of these, they actually have something called clades of HIV-1. Clades are different variations of it, and so it's a very diverse virus. Clade B, as an example, is typically America, Europe, is the majority of HIV cases. Clade A and C is Africa and Asia, and they are genetically different, which poses problems if we try to create a vaccine. Which clade do we target? What's going to give the most benefit? And I'll talk about that later as well when I talk about vaccines. So there is a lot of genetic diversity in this virus. So if we look at America, these numbers represent how many people per 100,000 of the population in that state. Georgia has the 8th highest rate of HIV infection. The top one is Washington, D.C., and it is considerably higher than everywhere else. It's mostly, as you'll see in a minute, around areas of metropolitan areas, big cities. So you've got a whole, quite a high load up in the New England area as well. What is interesting is that in America, of all the people who have been infected in the past and are currently infected, about 55% of those have died from HIV already. So it's got quite a high mortality rate. It may take some years, but it's still going to kill you one way or another. The next picture I'm going to be showing you is going to be a time-lapse series from 1983 with a yellow dot representing 50 cases, and you can see how it's spread across the country. This is in 1983, and you'll see it's really only identified around San Francisco at the moment. It's going to go every 2 years forward, and the more dots which appear will show you the more places it actually is. So we're having a spread around Los Angeles and California. I think that's New York area up there. You can see Atlanta here is getting more and more. And it's really sticking to the main cities as to where most of the problems are. And this is 2007, the latest data I have for this map. But you can see, you pick any of the big cities, I think one of these two is Dallas. I can't say I know geography that well in America. I grew up in England, so I'm struggling with, I'm not even sure, is these North and South Dakota? But the more rural aspects of the country have a lower infection rate. It seems to be mostly concentrated around the big cities. So if we take a look at the different groups of people who are infected to different rates. So if we're looking at the US population, this is just from the US Census information, and approximately 66% of the population is white. However, when we look at the amount of people with AIDS, so this is when AIDS has really taken over the immune system, you'll see almost half of it is in African Americans, but that only represents about 12% of the population. And there's a number of reasons why. There's that first reason, portrayed as a white gay man problem. So the African American community didn't really have any worries about it in the 80s, so it helped to spread it faster through the population. And then there's been a number of other risk factors suggested. And one of the biggest ones suggested is the socioeconomic status, which is also linked to the level of education. And that can also be linked to your access to health care, lower economic status. You don't have as good a health care plan, or maybe not one at all. So that can all come to problems. And when we're looking at that, without the level of education on how to prevent it, it gets spread faster. Some of the statistics also suggest that the African Americans generally have a higher number of sexual partners in a shorter number of years. And it's also suggested to be more high-risk sexual activity, more unprotected sex. So there are more reasons for why it's currently affecting the African American population as much as it is. It's not going to be any single one of those reasons. Probably going to be all of them combined together somehow. One of the other things which was linked was in the 80s. Apparently there was an increase in the drug use among the African American population. And sharing of needles would certainly spread the virus. So that was also one of the other possible reasons suggested. So Georgia. Let's see what Georgia has for us. It's 30% African American, 57% white. However, for the HIV AIDS cases, this is including HIV infected but not having AIDS, 78% in the African American population. So it's a huge problem. In the South, you see more in the African American population than elsewhere. If we take a look at comparing what's happening around the world to what's happening in America, to what's happening in Georgia, we can see that there's about 7,400 people per day being infected with HIV around the world. That's a huge number every single day. And about 5,500 people dying per day due to HIV or AIDS. In America, you're looking at about 154 people per day and about 63 people per day dying. And with Georgia having the 8th highest rate, you're looking at about 5 people per day being infected in Georgia alone. And The only data I have for the deaths in Georgia is a little bit older, from 2006, and that says about 2 people per day dying due to HIV. And the majority of them, probably around 60 or 70% of them, are in the Atlanta, Georgia area. So you're looking around Atlanta and the counties around there, where most of the infections are. So how can you be infected? How can you prevent yourself from being infected? Well, the main infection rate is sexual contact. You can also, if you're a mother and you're infected, you can pass it on to your child. You can cross the placenta in the womb and pass to it there. This one is not just across the placenta in the womb, it can be during birth, it can even be from breastfeeding. Virus has been detected in milk, so it is possible to infect the infant that way. And one of the main problems in the westernized world, America and Europe, is contaminated blood. This caused huge problems in the 80s before they identified the virus. Blood banks were contaminated with HIV. Have a transfusion in that time, then it was possible to be infected that way. Some parts of the world, if you go to some parts of Africa, their blood banks are still contaminated because there's so many of their populations infected, they wouldn't have enough blood. So be careful where you choose to have surgery at a cheaper price. Same thing for organ transplants. Same problems there. So anything which has contaminated blood is a way of infecting you. And we can actually reduce the chance of being infected if we modify our habits. And in the 80s, the homosexual community modified their habits, reducing their number of sexual partners, using protection. And that's the group which had the biggest reduction in new infection rate, just by modifying their habits. Heterosexual contact, use protection, and you can reduce your chance of being infected. So there are fairly easy ways to try and reduce your chances of being infected. We screen the blood now for HIV, so that doesn't go into the blood banks. Chances of being infected depends on how much virus is there. The more you have, the greater chance you're infected. If you've got a single virus going into you, it does not necessarily mean you will be infected, but you're never going to have a single virus going into you. In any of the bodily fluids, you're going to have hundreds, thousands, or more of the viruses present. The different routes of infection, it depends whether you're male or female. Female, your biggest problem is high-risk heterosexual contact. That means unprotected sex or multiple sexual partners at the same time. If you're male, you're looking at homosexual contact as the biggest route of transmission. The drug use is increasing. It's been one of the ones which has increased over time. And when you're looking at places like Africa, the drug use is not necessarily as high. And we'll look at the global methods shortly. If we compare globally, majority heterosexual contact. And this is because of Africa having 2/3 of the HIV cases. Whatever is happening in Africa will influence the biggest route of transmission. But more of the homosexuals, it's a higher proportion of the homosexual population who are infected, so that's why more men are infected that way. But men and women are equally likely to be infected through high-risk heterosexual acts. So America, one of the biggest problems is homosexual activity. Globally, that's a very minor problem, really. It's mostly heterosexual activity. And that comes down to Africa again. They have most of the cases, and that's the biggest transmission route there. So the virus itself, well, it's actually not a very strong virus. It doesn't survive very well in the environment. If it dries up, it's basically dead. If it does get outside the cell, it can take, they estimate, up to about 6 hours for air droplets to dry, depending on how big the droplet is. And you can quite easily kill it. You can use basic bleach at the recommended dilutions. Kill it. You can use what we use in labs, 70% ethanol. We'll kill it. And you can also put it at different acidic or basic ranges, and it will be destroyed. So it's quite easy to kill it when it's outside the body. It doesn't survive very well. So it's said to be a fairly weak virus in that respect. So the virus itself, this is what it's supposed to look like. It has its genetic material surrounded by protein. That protein protects it. And then it actually has an envelope on the outside. And this envelope is basically part of your cell membranes when a virus is released. That's where it comes from. But it has these protein on the surface. Actually has 2 of them. And used to allow the virus to bind to its next target cell and infect that cell. So I'm going to go through what it does for one cell first, and then I'm going to go through what it does for your whole body over time. It's important to understand what it does to your cells to realize what it can do to your body. So when we're looking at this, we'll just take one virus coming along to your general basic cell. It's going to come along and it has to attach. It has to bind to the cell. It binds to its main receptor is CD4, and that's a receptor on your immune cells. It's actually on a specific population of immune cells, a subcategory of your T cells, which are called T helper cells. Those cells, the T helper cells, control your immune system. They direct it, they activate it, they make the cells work better when they tell them to work more. And without those CD4 cells, your immune system really does not work well at all. It can't fight those opportunistic pathogens, which I was talking about earlier. And this virus will kill those cells. So but when it infects them and eventually kills them, it's going to destroy your immune system. So you cannot fight normal infections. So once it's bound to the cell, it has to get in. So it goes through a process called uncoating. Basically, though, the lipid envelope of the virus fuses with the plasma membrane of your cell, and the virus enters, the protein part enters. And once it's entered, it goes through a process called reverse transcription. The virus has a genome of RNA, which is, we have DNA as our genome. And so it basically takes that RNA and turns it into DNA. And when it's turned it into DNA, it sticks it into your chromosomes. And there's no way of removing it that we know of after that. And it can sit there for many years doing nothing, but it's still in your chromosomes. And it may be activated later on, or it may be activated very quickly. No one knows what causes them to be activated. There's probably a number of different factors involved. But this is why it can be so dangerous, because it can just sit there in your cells forever. And then it can just come back at you at any point. There's no way of identifying this cell at the moment that it's infected from your immune system's point of view anyway. So it can sit there for a long time, or it can start to make copies of itself. When it's like this, it's said to be dormant, and then it becomes activated when it starts to make copies. When it makes copies, it's going to copy its genetic material, the RNA. It's going to make some viral proteins. During this, you use viral proteins to cut them up and make them the right shape so it can start to form the viruses. Then its second generation can go on and infect many more of your cells and other people's cells. And if this is just one virus coming in, it can continue to release these indefinitely. So you can have hundreds, thousands, millions of viruses produced from a single cell, depending on how long that cell survives. Also, if it's in your DNA, if your cell copies itself, replicates itself, it's also copied the virus. And all of the offspring of that cell will also be infected. It puts the DNA into your DNA, and it makes it part of your DNA. And then that cell will always have it. So if you can't identify that cell and kill it, you've still got a potential source of more virus being produced. So you can test for it and find it, but your immune system cannot. So in a lab, we can actually look to see if this DNA is inserted in your cells or not. Okay. Take some of your cells and have a look and see if we see it or not. But your immune system has no way of doing that. So the different stages of HIV infection, there are 4 clinical stages, but often they're referred to as 3 phases. And phase 1 is stage 1 and 2 combined. Phase 2, 3, and phase 3 is stage 4. It's easier to look at the infection on these 4 stages rather than just 3 phases. That's what I'm going to take you through. The first stage, this is when you become infected and how your body reacts to that infection. Second stage, this is where you have a virus, you're producing virus at a low level, but you don't have any signs or symptoms that you're infected. You may never know you're infected at that point if you don't get tested. Third phase, your immune system starts to fail, becomes slightly weaker and weaker. And in stage 4, that's when your immune system's been destroyed to the point where it can no longer function correctly. So we're going to have a quick look through these, and we're going to follow a couple of different things. The red line, which comes down and goes back up, that's your CD4 cells. Those immune cells that are going to control your immune system. And you're looking at how many are present in your body over time. The black line, which is high to start with and then comes down, that's how many viruses are present or detectable in your body. So in stage 1, this is this little stage here. It only lasts between 2 and 10 weeks on average, but there are some variations from person to person. And you think you have a flu. So how many of you go to the doctor if you think you have a flu? How many of you stay at home and just don't do anything? That could still be HIV infection. You have no way of telling the difference between them. So at this point, you don't know you've been infected. And this is when your immune system starts to respond to that virus, which comes back to your question earlier. It does take probably around, I think it's about 6 weeks on average for your body to start to produce antibodies against the virus. So in those first 6 weeks, there's no way of identifying if you're infected or not. So if you do give blood, then you could contaminate the blood supply. So that's
6 weeks for, sorry, 6
yeah,
months?
6 weeks. Yeah, it's about 6 weeks, not 6 months.
Okay.
Yeah, so it's on average around, I'd say mostly 4 to 6 weeks when they start to produce the antibodies. So that's the first way of telling you're infected. And when you do start to produce antibodies, you're said to be seroconverted. Seroconversion just means your body is now producing antibodies against whatever it is, in this case, HIV. At the same point here, your CD4 count drops because it's infecting all those CD4 cells. But then you have a recovery of the CD4 cells in this stage. They come back to near normal levels. Not completely normal levels, but they increase in number again. And that could be due to your immune system responding to the virus at that time. In stage 2, your number of CD4 cells starts to decrease. So your immune system is becoming slightly weaker each day. However, it can still handle the majority of things you come across. So you're said to be asymptomatic. You don't show any signs of problems. Your immune system still seems to be functioning correctly. And this can last any number of time. Some very short, low number of years. Some people it can be very long year. A decade, 2 decades maybe. Anything's really possible. Depends how many ones go dormant and how many are active. And when you're looking at how much virus is here, if you start to see more and more virus appearing in your blood, then it means it's defeating your immune system faster. So it's a good prognosis indicator how much virus is in the blood. And this is when you infect everyone else. You thought you had the flu and recovered. You don't know you're infected. you go and infect other people with this. This is how it gets spread around without people knowing they're infected. Stage 3, this is when your immune system has got to the point where it's not functioning as well, so you start to get some opportunistic infections occurring, opportunistic pathogens being able to overcome your immune system. And you can have various different types. You have bacterial. Some of the more common bacterial ones could be Treponema pallidum. Anyone know what that causes? That's syphilis. Treponema pallidum. You also can have more mycobacteria. Anyone know that one? TB, tuberculosis, caused by a mycobacterium. Also leprosy as well. Some of the more common viral ones at this point, Kaposi sarcoma, that cancer caused by that herpes virus. Human papillomavirus, that causes warts. You can also have herpes simplex virus, more cases of that, cold sores, genital herpes. Protozoa, that would be that pneumocystis, causing pneumonia. And the fungal one would be Candida. That causes thrush, also causes vaginitis. So these are the ones which your immune system typically can control, and these are the ones when your immune system's not functioning as well, start to appear. When you go into stage 4, this is clinically defined as having less than 200 CD4 cells per mil. And at that point, your immune system is basically ineffective. It cannot fight anything. So you get a lot more opportunistic pathogens coming in. The ones which you picked up in the stage 3 start to take more control. And they spread even faster and cause more signs and symptoms of disease. And at this point, anything which normally infects you could be an opportunistic pathogen. Anything. A common cold could be a lot more severe. And it's at this point the amount of virus in the blood, these two black lines, starts to increase as well. So you can spread it more easily. At this point, you're not only having more infections, you're also having your lymph nodes will swell up. The lymph nodes are where your immune cells hide and work. So if they're trying to fight more pathogens, they're going to get bigger. So you might have swollen lumps on your neck as an example. You're going to have shingles. Who's had chicken pox? Most people will have chicken pox. And that goes and hides in your neurons, in your nerves, in your body. And at some point later on, it may or may not come back as shingles and cause a secondary infection that way. So you'll see more cases of shingles appearing here. We also have more generalized high fever, night sweats appearing. So you're generally not doing very well. Any questions about these?
In all these stages, there is no antibodies being produced at all?
No, there is. So you start to produce them once you seroconvert in stage 1. But those antibodies are all ineffective. They don't do anything against. They can bind to the virus, but they don't stop the virus from working. No, those antibodies, when they bind to the virus, the ones your immune system generally makes, don't do anything against the virus. It can still go and function normally. They offer no protection. And that's just due to the structure of a virus, which was more detail than I was going to go into here. So how can you diagnose it? Well, you can look for those antibodies, but you have to wait for seroconversion to occur. Cannot detect those antibodies if seroconversion has not occurred. This is the main way of looking for HIV, but you can never rely on a single test. If it comes up positive for antibodies, you have to do a second, possibly a third test to confirm that. And those tests have to be something other than looking for antibodies. And the reason I do that is because initially there was a problem with the specificity of the HIV antibody tests. Some antibodies out there which bind to other viruses were initially picked up as HIV antibodies, even though they weren't. And that will give you a false positive. Those tests are becoming less and less common, so this is less and less of a problem. but it can still occur from time to time, where an antibody looks like it should be binding to HIV, so they say you're infected, but in actuality you're not. So they always perform a second test before they tell you anything. They don't want you to panic for no reason. And with other tests, you can look for the virus itself, or parts of the virus in the blood. You can look for some of that DNA inside your immune cells. Take a sample of blood and see what virus is in the blood, what's in the cells in the blood. And you can also detect the whole virus in the blood as well. These are a lot more definitive identifiers that you are HIV infected, because you're looking for parts of HIV or the whole HIV particle itself. But these ones are much quicker and probably cheaper to do. Any questions on the diagnosis? Some of the genes in HIV are unique to HIV. That means the proteins which come from those genes are also unique. Common one is P24. That's a protein, it's one of those protein shells surrounding the genetic material. That's unique to there. We don't make that or anything similar to that. So if you look for a unique one, then yes, you can use it to diagnose. No, when you're screening the blood supply, they do 2 or 3 of these. When they're testing you, they do 2 or 3 of these. You can never rely on a single test when it comes to HIV, because it's such a devastating disease. So, antiviral therapies, these do not cure you. They only slow down the virus, but the virus will still win eventually. So once you're infected, then, and you take these, you're just prolonging the time until you die from HIV or AIDS. No, because it's destroying those cells which control your immune system. So those CD4 cells, they direct the whole of your immune system. Without them, your immune system cannot really function. And if it's killed those cells, when you get AIDS, it's down to less than 200 per mil, where normally you'd have probably over 1,000 per mil. Then no matter what you do to the immune system to make it work, it can't be directed, then it's not going to do anything. That varies from person to person. It depends on your specific immune response. It also depends on whether you're taking antiviral therapies or not, which drugs you're taking to try and slow it down. And there's a number of other factors no one truly understands as to some people. It can take decades. The people it can take 1 year or less than a year. So no one really knows what makes it become active or why. I'm not going to go through the actual names of the drugs. I'm just going to go briefly through what the drugs actually target. So first of all, you can try and stop the virus from entering your cells. If you stop it entering your cells, you stop the infection process. And there's a couple of drugs out there which are used for that. You can target reverse transcription, turning that RNA into DNA. And there are 2 different types here. You have nucleoside reverse transcriptase inhibitors. These are basically DNA analogs. They look like DNA, but the virus enzyme is not as specific as our enzymes. And so when it uses it preferentially uses it instead of your body, it actually stops the DNA from being converted properly. Yes, some of your cells will take it up and use it, but the virus enzyme will take those ones up a lot more easily and faster than your own cells. You have non-nucleoside inhibitors. These actually target the protein enzyme and stop the enzyme from functioning. So 2 different ways of targeting that. We don't have anything similar to reverse transcriptase in our body. It's unique to retroviruses, so it's a good target. We also don't have anything to integrate DNA into our DNA, so we can target the integrase enzyme in the virus. We've got several drugs against that one out there. When the viral proteins are produced, they have to be activated for the virus to become infective. And one of them is through a viral protease. We don't make that protease, only the virus does. So if we target that one, and there's several out there, we can stop the ones being released. They won't be active. They'll be non-infective. And then we can also target the process of the virus being released. Our cells don't naturally make viruses. There's a number of parts in there we could target for that as well. But we never take just a single drug. We use combinations. And that's because HIV mutates and it becomes resistant to these drugs. So if you're just using one drug and a virus picks up a mutation, it no longer works against it, that one mutant will then release hundreds, thousands of viruses which will go and take over all your other cells in your body. So it will take over the population. If you target 2, normally it's 3 different parts, you have less chance of a mutant being able to occur for all 3 at the same time. So it reduces the chance of mutation. These antiviral therapies, they do have problems associated with them. There's a whole long list on the screen. And this basically makes some people skip their medication or stop taking it. And this is a major problem. If you miss a single dose, then you increase the chances of a mutant to that drug appearing. And when you just stop completely, then anything which was slightly mutated can evolve a little bit faster, and those drugs no longer work. You infect someone with a mutant virus which is resistant to the drugs, they can no longer use those drugs to treat them. So it's a problem with the resistance spreading through the population, which is why if you're going to take the drugs, you take them on time, every time, all the time. If you decide you don't want these effects, don't take the drugs and make them useless for everyone else. The mutation rate is very high. In HIV, it's approximately 10 bases per replication cycle. That's equivalent to 3 amino acids on the whole of a virus changing every time it replicates itself. And that is a tremendously high mutation rate, which is why it can mutate to those drugs very easily if you skip medications or stop taking them. And this is what causes those drug-resistant strains to appear. I've already talked about why we use many at a time. Vaccines against this, a lot of research in this area. First of all, vaccine is used to stimulate your immune system to be able to protect you when you come across the natural pathogen. So for the influenza, if you have a swine flu, you can either have the nasal shot or the injection. Either way, it's basically telling your immune system to look out for this. And when it next sees it, it can mount a faster, larger response against it. And the ways to do this, you can either target the virus itself, stop that virus from being able to infect your cells. That's when antibodies bind to the virus and block it. And find HIV, don't do anything against it. Or you can target those cells which are infected. If you can find a way for your immune system to see that it's infected, and you use a vaccine which will induce your immune system to attack all your infected cells, and you eliminate them before you destroy your immune system might be able to repair itself or build up more of the CD4 T cells. But you have to have a way to identify the infected cell. I'm going to talk about the 3 major vaccine trials which have gone ahead for this. And vaccine trials have 3 phases. Phase 1 is a very small one. And all you're looking for is this safe to use? In phase 2, you take hundreds of people, possibly in the low thousands, and you're again testing the safety in the human population. And recently they have started to try and see, are we seeing any positive results? But you can't really do that on phase 2 trials because there's not enough people there to give you enough data. Phase 3 is when you finish the safety testing. You still continue it always anyway. But it's really, phase 3 is to test, is this vaccine working? Is it effective vaccine? Does it prevent people from being infected? Why it's hard to make a vaccine against HIV? No one's ever recovered from being HIV infected. Not even Magic Johnson, which is what everyone asked me initially. He's still HIV positive. He's one of those people where it's taken a long time. Maybe it's because he's got lots of money and he can afford the best health care and the best antiviral drugs, prolonging the time. HIV, as I've said, destroys your immune system. If you destroy the immune system, how are you going to get it to work for you afterwards? It puts that DNA into your cells and hides. How do you identify that infected cell? Even if you vaccinate yourself, what's to stop this coming back at you 10, 20 years down the road when your immune system's forgotten about it. I mentioned those clades earlier. They're very different to one another, genetically unique. They're mutants compared to each other. So which one do you target? Do you target the most common one in Africa to tackle the biggest population? Or do you target the one in America and Europe, which is probably the smallest population? But they're the ones who can afford it and put the research into it, which is where most of it's coming from. And even within each clade, each subtype, there's still a lot of variation. It might not protect you against all of them. The last problem is that even if we did come up with a potential vaccine, we can only test it in humans, because there's no good animal model to tell us if it would work or not in humans. As I said earlier, the SIV1 viruses, they have no effect on the monkeys really. So you can't use them as models to see if your vaccine's working. So the three main vaccine trials, the first one, Aidsvac. This one targeted an envelope protein on the virus, and it's trying to stimulate an antibody response, which would bind to the virus and stop it being able to infect your cells. And there were 2 different trials done on this, but there was no effect. There were no antibodies produced, which prevented people becoming infected. Yes, it did make antibodies against the virus, but they bound and they did nothing to the virus. So we bound there and the virus can still go on and continue its life quite happily. So this one failed. The second one was started around 2004 or prior to 2004. And this one was trying to target your immune system to recognize infected cells. So if you can't make antibodies against a virus, make sure your immune system can see those infected cells and destroy them before they cause you any harm. They stopped because it was termed that the vaccine wasn't doing anything. You were equally likely to be infected if you were vaccinated or not vaccinated. So the trial stopped. They didn't inject virus into people. That's an ethical problem. When you're doing these, you have to give the vaccine to high-risk groups and give people in the same high-risk group a placebo. Placebo is basically you inject some water into them or something like that, and they don't know whether they receive the vaccine or just a placebo. And then you compare the 2 groups later on to see if the vaccinated group was more protected, had a lower infection rate compared to the placebo group. But at the end of this trial, when they looked at the data, it was about equal. The vaccine offered no protection, so it stopped. In this one, which is trying to make your immune system attack those infected cells and get rid of them, the step 1 was the first to start. The Fambili is the exact same vaccine as the step 1, just in a different population. And when they did this, this vaccine actually made it worse. The vaccine did produce an immune response, but the immune response allowed the virus to enter your cells more easily. So it actually increased the infection rate. As soon as they saw that, they pulled the plug on the trial, and they also stopped the second trial of it, which I think was about to be started or just started in Africa. So this is where the vaccine was actually worse than the placebo. It increased that infection rate. The third one, which only just recently ended, was Alvac. That's the new part of it. And that targets your immune system to target those infected cells. But it's also, they threw that old Aidsvac one in there at the same time to see if using both would increase the chances. So not only targeting your own immune cells, which are infected, but targeting the virus at the same time. So you're trying to attack it on 2 fronts. This one was a huge trial in Thailand, and initially, when they reported this data, they showed there was around about a 31% reduction in the new infection rates, which was occurring. However, they didn't do a proper analysis, and they didn't give the data to anyone to analyze either. When they reanalyzed it and looked, it only showed a 24% reduction in the infection rates. And statistically, that is just due to chance. No, they had 16,000 people, but 24% reduction could just purely be chance when you're looking at vaccine trials. It's still considered statistically just chance alone. So this one is also not an effective vaccine. And the trial has ended. They're still analysing the data. I'm not sure the Thai government has released all of the data yet. So this was only in October when they announced their initial results. And it was October, November when they confirmed that no, actually really didn't do much. For a vaccine to be effective, you need it to be probably at least 60% effective. Probably higher than that, the higher you go, the better it is. The problem which this vaccine trial created was a media hype about we may have an effective HIV vaccine. People started to consider, do we need to continue these expensive prevention programs? And word of mouth spread that there's a vaccine out there, so you can go and do what you want. And that's considered to be an upcoming problem in Africa. It's been spread around Africa, word of mouth, and the programs of prevention are being reduced because they think the vaccine worked. So that's a major problem which could halt that reduction in new HIV infections, 17% reduced since 2001, could start to go back up again. So you have to think about when you take results, when you release them, and what effect it's going to have. These are the websites where you can get more information. This top one, avert.org, is an excellent website for information about HIV. These ones here, they are very good websites, but sometimes it's difficult to find the information you want.